Good. Okay, so this will be the overview of the talk. So I'm going to give you a brief introduction on hippocampal, on the episodic recollections and hippocampal sharp wave ripples. And then I present the main method that I use in my PhD, which was intracranial EEG recordings in patients. And after that, I'm going to go through the main results in 3 different studies and leave time for discussion and summary in the end. Okay, so to begin with, I want to start with a few words on episodic memory retrieval. So for that, I'm going to present this short video taken from the Pixar film Ratatouille. What you're going to see here is the food critic sitting in a restaurant and tasting the food. And after he takes the first bite from the food, then suddenly childhood memory comes to his mind. And what is so nice about this animation, I think, is that it captures so nicely what like how immersive the episodic recollection can be, and also how diverse and vivid is the mental content that is represented in our minds during this such recollection. When I am talking about episodic memory, I'm referring to a recollection of a specific personal experience that is often situated in an explicit spatiotemporal context, but not necessarily. And usually these memories involves multimodal information that are represented broadly across the cortex. And during the recollection, there is a process of reenactment in which the neuronal representations that were engaged during the original experience are reappearing and re-emerging across the brain. And this is believed to be the process that enables us to experience, to re-experience, and to feel the past in such a vivid format. And when you retrieve such complex memories, for example, as the one that presented here, then what you have is a prolonged orchestrated replay of an entire scenario in your mind that includes so many different contents and representation. And I think this process is quite remarkable, and it's a really great, like very deep question, how the brain does this, how the brain is able to orchestrate and replay in such an ordered and coherent manner all these different representations. For example, the spatial configuration of the kitchen, the look on his mother's face, her voice, and the sounds of her washing the dishes, and the taste of the food, the smell of the food, all of this coming back to our mind when we recall something in a very organized manner. And I think this is the basic question that I'm asking in my PhD. And the immediate suspect that come to mind when we think about this mental process is, of course, the hippocampus, which we know back from the 60s that from the 50s and 60s that it plays a major role in declarative memory in humans, and selective damage to this region in the brain is associated with a selective damage to our ability to form new episodic memories. But not only that, but also after damage to this region, patients usually suffer from retrograde amnesia, which in fact affect their ability or impair their ability to recall also existing memory up to several years before the injury. So this brain structure is important for recollection as well. However, what is the precise mechanism in this brain structure that actually support the recollection and perhaps orchestrate all these wonderful reactivations that we get during recollection? And this remains to be open, and this was the question that I investigated. So what are sharp wave ripples? Sharp wave ripples are first brief high-frequency LFP oscillation that emerge in the CA1 subfields in the hippocampus. What you see here is the coronal slice of the human brain, and enlarged in here you can see the hippocampus, the human hippocampus, with its different subfields. Okay, and right over here is the CA1 subfield where ripples are generated, and and you can see this place here is actually the main station where the output of the hippocampus goes out to the rest of the brain, to the cortex, either directly through the CA1 projection or through this subicular entorhinal output pathway. So this oscillation, why are these oscillations so interesting? First of all, because it was shown to be conserved across a variety of species. species, sorry, from rodents to monkeys and humans, all of them show this transient bursts of activity, transient oscillations in the hippocampus. And if you zoom in to the hippocampus and examine what is the activity in the hippocampus that emerged during these oscillations, what you see is that 10 to 15% of the pyramidal neurons in the hippocampal and neuronal output pathway fire together. And this, I think, research here from Yuri Buzaki's group illustrated really nicely. What they did, they recorded more than 50 different neurons inside the hippocampus, together with the hippocampal LFP, and they detected ripple events. You see here the black circles. And what they found is that every time there is a ripple, there is a sharp increase in the synchrony between neurons. So you see here within 100 milliseconds, you see many of the neurons emit a spike. Now, if you look at this activity from the single unit level, you don't see anything special. You see a spike here, a spike there. However, once you zoom out a little bit and look at the entire ensemble of hippocampal neurons, then you immediately see that something very dramatic happens here. And the hippocampus actually sends a major output during these ripple events to the rest of the brain. Now, the first study that demonstrated very convincingly that the ripple events in the hippocampus indeed affect the rest of the brain is the study from Logothetis et al. In 2012. What they did is they recorded, using an MRI-compatible electrode, they recorded the LFP in the hippocampus and detected many sharp wave ripple events, hundreds and thousands of them, while the monkey was awake inside the magnet, inside the MRI scanner. And this allowed them to compute the peri-ripple or the ripple-triggered bold activity across the brain. And what they found, first, that inside the hippocampus, you see a significant increase in bold activity, time-locked to the ripple events, here in time 0, and you see here in seconds. We're talking about a rather slow activity because this is bold signal. But this is expected because we saw that there is an increase in firing rate during the ripple. However, when you look at the entire cortex, you see that major portion of the cortex are also activated in a way that is time-locked to these hippocampal events. And interestingly, regions in the subcortical regions and also early sensory regions show the opposite effect of deactivation coupled to the ripples. So obviously, we're talking about an event in the hippocampus that has a major influence on the activity across the brain. Now, what is the function of ripples? Okay, so very exciting studies in rodents that have made in the past 10 years have shown various results about this. So first of all, there was studies found that there is a causal link between spatial learning and memory in rodents and these hippocampal events. This means that if you interfere selectively with the sharp wave ripple events in the hippocampus, you impair the memory of the animal. And conversely, if you enhance this oscillation, this spontaneous oscillation that emerge while the rat is navigating inside a maze using optogenetic stimulation, then you actually improve the memory. Okay, so there is a causal link between ripples and memory performance. And also, another line of study have shown that these events are critical for memory consolidation and for memory reactivation during slow wave sleep. So if you interfere with ripples during the slow wave sleep, you actually impair the memory as well, and that's it. Now, however, regarding their role during the waking state, the picture is a bit less clear, and we are still not sure whether they have any cognitive role or what is the content that is attached or associated with these events. And this is mainly because it's quite difficult to assess the detailed mental content that an animal experience during these events. So you cannot ask directly the rodent what it thinks about when the hippocampus elicits a ripple. However, in humans, it is possible. And this was exactly the motivation behind my research. I wanted to examine the cognitive role and the mnemonic content of awake hippocampal ripples and characterize the nature of the hippocampal cortical interaction during conscious reportable recollection in humans. And to do that, I used a very unique and rare method of invasive intracranial recording in patients. here you see example of these signals that were recorded. These patients suffer from pharmacologically resistant epilepsy, and they come to the hospital in order to do invasive intracranial monitoring of brain activity, in order to using this depth electrode and the subdural grids that are placed right on top of the cortex, or penetrating the cortex and recording from hidden places in the cortex or from deep structures. So using this invasive electrode, now the clinicians can monitor this brain activity 24 hours for several days and wait for several seizures to occur. And after that, they can pinpoint exactly where is the focus of the seizure and perhaps treat it using a neurosurgical treatment by disconnecting, for example, the pathological region. Now, during the waiting period, when the patient lay in their bed in the hospital and wait for seizures to occur, there is a unique opportunity to actually look at investigate the brain activity in individuals who can actually interact with you, the awake individual who can interact with you, and also can report about their mental content online. And this allowed me to actually investigate what happens in the brain, and especially between the hippocampus and the cortex, when a dormant memory comes into mind. And this was this is what I'm going to show you next. So just a few more details about the intracranial recordings. So I'm going to present you 3 different studies. 2 of these studies were done in New York, in the North Shore University Hospital, and 1 of the study was done in the Stanford University Medical Center. We had recording for a relatively large cohort of patients. About, you see here the numbers, 50, 20, and 12 patients in each study. And in each one of these patients, we actually record from multiple recording sites. Okay, so up to 300 individual recording sites in each patient. This is, and importantly, we have also both recordings from the simultaneous recording from both the hippocampus and cortex. So it allows us to actually investigate the interplay between these 2 structures. So now briefly about localization of the electrode. So before the surgery, patients undergo an MRI scan for anatomy, and after implant, they are entering into a CT scan, and which visualizes the place of each individual contact. And once you co-register the 2 scans together, you can identify very precisely where each electrode sits, and you can reconstruct the brain and with a very precise anatomy of where the electrode is situated. And after integrating data from several patients, you get these beautiful maps. We use a surface-based alignment to bring all the subjects to the same common space. And you see that we get a pretty wide and broad coverage of electrodes across the cortex. Each black dot here is 1 recording site, okay? And importantly, we have a lot of electrodes in the inferior temporal cortex, where high-order visual representations are located, presentations of specific visual objects such as faces, outdoor scenes, and objects, etc. And this will be very handy soon, you will see. So now, just to show you the depth electrodes that are implanted in the hippocampus. So we're talking about this kind of electrode. They penetrate the hippocampus from lateral to medial, crossing several subfields within the hippocampus. And importantly, what we are interested is in the contact that sits just in the CA1 region, where ripples are generated. And this is what we investigate. So now for the first study, this is the task that we use. We use the visual free recall task. The patient begins with a short resting state period to have a silent, clean baseline. And then we present the patient with 14 different pictures that belong to 2 different categories. Either popular faces, such as Barack Obama, Jerry Seinfeld, etc., or popular places, such as the Eiffel Tower or the White House, etc. And the picture are presented 4 times in pseudo-random order to give enough opportunity for the patients to actually memorize the pictures and remember them in great detail. And then after a short distraction task, the patients are required to freely recall these images. However, we tell them to first recall all the faces they could remember, and then all the places they could remember. So they do it in 2 separate blocks. And we run this task twice in each patient. So in the second run, we reverse the order of categories. And importantly, during the free recall task, the patients are blindfolded. So we completely block any external visual stimulation in this patient. And we also record the voice recording during the free recall using a microphone attached to their shirts. And now, this is the example of the stimulus, of the stimuli that we use in this task. So we try to select very vivid, colorful images that are full of visual details. And during the free recall period, we we asked the patient to not only name the pictures they recall, but also to describe them with 2-3 prominent visual features. Thereby, we're ensuring that they retrieve also specific visual information about the pictures, not just general semantic details. And now for the results. I'm going to present the results in two parts. First of all, I'm going to begin with the hippocampus. I'm going to show you sharp wave ripple activity in the hippocampus during this task. And after that, I'm going to discuss how the hippocampus, how the ripples affect the cortex by analyzing the peri-ripple cortical activations in the cortex. So now, just a few words about ripple detection. So as I told you, we are focusing on the electrodes that are located in CA1, and we extract from the raw LFP, we bandpass the signal between the ripple band, and then we extract the envelope and normalize it, and then we detect all transient events that cross for standard deviation. And you can see here the overall average wavelet spectrogram of the ripple events across all patients and all events during the task. So you see that you see the typical spectrotemporal signature of the ripples with peak frequency around 90 Hertz. This is compatible also with previous studies in humans. And now I want to remind you just what are the 4 main experimental conditions that we had. So we have the resting state period, which serves as a clean baseline. And then we have the picture viewing stage, which is the time where the memories are formed. And then we have the free recall period, which can be subdivided into 2 different conditions. The actual recollection moment, when the patient is actively describing a particular image, and the memory search period, in which the patient attempting or trying to recall a particular image from an image from a particular category, but failed to come up with a specific item. So we call this memory search. And now for the results. So I'm beginning with the picture viewing results, picture viewing stage. So the analysis that we did is we computed the peri-event time histogram of sharp wave ripple events, aligned to the onset of the picture on the screen. And what we found was the following. We found that when the pictures are presented for the first time, we see a general increase in ripple rate that peaks about 700 milliseconds after picture onset. During repeated presentation, as you may see, we still see a lot of ripples occurring. However, when you look at individual items, you see that some of the items produce a high ripple rate, other items produce a rather low ripple rate. And when you average all of them together, then you get this flattened response. So to deal with that, we use the median split, and we took the upper half of the images and the lower half of the images in each patient. And this is how the response looked like when we separate these 2 groups. And now the interesting question is what happened during free recall when the participants are blindfolded and recalling spontaneously, recalling the same images. And what we find is the following. We found that second before that ripple rate is significantly increased, peaking a second before the onset of verbal report, only when the participant recalled these same images that also produce a high ripple rate during the picture viewing stage. So now, just to demonstrate this result, I want to show you this video clip that this movie that I prepared, and you will going to hear the ripple event like sound like this sound, and I'm going to mark them also with with yellow triangle, and you will hear different patients spontaneously recalling images while their eyes are closed, are covered.
Oh my God, sorry. Um,
Oh, sorry, why is this stuck? 1 second. Okay, yeah, now notice.
what right now? Oh, it's the White House with the fountain
So
that
I
looks
wanted to
like it had
notice,
green
give
water.
you another example. I wanted to notice that just before the patient is recalling the image.
There was the girl from Friends. She was
The
playing
hippocampus
on the
elicits this ripple
guitar
event.
with the blonde hair, long blonde
Just one
hair.
more example.
What else
Wait
did
for
I
it here.
see? Oh, Leonardo DiCaprio holding up a martini glass or champagne glass. His blue eyes were
And
really
quiet, no
noticeable.
ripples. And then the next recollection
Mm,
and some
Uma
additional ripples
Thurman
during the
smoking a
recollection.
cigarette, like red
Okay,
lipstick, short black
so
hair.
this I think demonstrated that hippocampal ripples, that during recollection, the hippocampus elicits sharper ripple events that slightly precede the onset of verbal report of recall. And now I think a very interesting question is what happens in the cortex during these transient hippocampal bursts. Okay, and I just to remind you, we have a lot of many cortical recording sites, and importantly, we have recording sites that are in the inferior temporal cortex, where we have category selective representations, visual representations. Okay, so this is, for example, one. Here you can see one recording site that was situated just in the fusiform face area and was completely, as you see here, selective to faces. Okay, so it almost didn't respond to places, but responds very robustly to faces. And now an interesting question is what happened in this inferior temporal site that are category selective when the hippocampus elicits this ripple. And this was exactly the next analysis. I took all the category selective visual electrodes in the inferior temporal cortex and other regions. And now you can see here the stimulus response in this electrode for the preferred images of each electrode and for the non-preferred images, preferred category and non-preferred category. And then we now ask, okay, what happened during free recall when the patients are recalling now images that either belong to the preferred category or to the non-preferred category? And more specifically, what happens in this electrode when the hippocampus elicits the ripple? Okay, and this is what we did here. So you see time 0 is the onset of the ripple in the hippocampus. And what we find is the following. We find a content selective peri-ripple reactivation of the visual cortex. This electrode showed a transient activation coupled to the ripple, but only when the patient recalled the category that is relevant to this recording site. Okay, when the patient recalled the non-relevant items, then these cortical sites remain rather silent during the ripple. So this demonstrates that the ripple event is coupled with reactivation of a memory representation in the visual cortex. Now, importantly, here you can see the spectrograms of this response. So you see that we indeed talk about a broadband gamma response, broadband high frequency response that reflect local increase in population firing rate. And you can also see that the activity in the cortex starts slightly before the hippocampal event. And this points to the fact that what we're talking here is not on a unidirectional signal flow from the hippocampus to the cortex, but rather we are talking about an interplay or a bidirectional interaction in which the cortex begins slightly before the ripple, perhaps biasing the hippocampus, biasing the activity in the hippocampus, which then generates a sharper ripple event and activates further the cortex with the relevant information. So I have a sense, because I don't have much time, I won't get too much into it, but I can talk about it later. but it's also very compatible with other studies in rodent showing this information, this bidirectional information flow. Okay,
But it's
yeah,
sorry for interrupting,
yeah,
but calling it bidirectional is a little bit kind of your wishful thinking for interpretation. In any other, these kind of pictures in any other domain would say input comes from visual to hippocampus.
yeah. This
I
is
don't know why you want to call it bidirectional or hippocampus to
okay.
visual. I see
So
one direction here.
yeah. So it's a great question, and I was, I did many analysis. First of all, I have another analysis here that showing precisely this, how during the actual ripple event itself and not before the ripple, you see that item specific, not category specific, item specific representation are activated at the peak of the ripple and not before it, compared to the category selective response. So this activity here may be it's less specific, more broad, than starting before the ripple, but then in order to activate the precise representation of the memory, the specific image, then you see this activity happens only during the actual window of the ripple and not before it. This is one evidence that I have. Other evidence come from rodent studies that show very nicely this bidirectional signal flow, and we can discuss this later. But just to keep in mind that it's not, at least it's not a unidirectional signal flow from the hippocampus to the cortex, and the question whether it's not a unidirectional flow from the cortex to the hippocampus, this we can argue later. But I have some evidence that show that it's indeed a bidirectional, and we can discuss this. And let's leave it for the end of the talk. Now, just to remind you, we also had this inter-recall intervals, this memory search period, where the patients are not recalling a particular images, but still targeting a particular category of images. Okay, and what we see is that there you don't see this content-specific reactivation of the visual cortex, which suggests that these sharp wave ripple coupled activations in the visual cortex are specific to conscious reportable recollections and not to just internal memory search. And here you see the statistics, and here this is the significance, marking the significant time period. And you can also see that during resting state, you don't see this cortical reactivation, this cortical activation. Okay, so just to summarize this part, what I've shown you is a link between conscious recollection and sharp ripple events. Okay, this is the first demonstration of this link in humans, and it's also a demonstration of the role of ripples in a non-spatial, non-navigational task. And this is important because it generalizes the role of ripples not only to the domain of spatial navigation, but also to other domains of declarative memory in humans. Second, I showed you that ripples seem to precede the onset of verbal report of recall. And this is interesting because it may suggest an early cortical hippocampal memory trace reactivation as a possible recall initiation or boosting mechanism. So first you get this hippocampal cortical representation activated, and only then you're able to become conscious of it and report it verbally. And third, I showed you that ripples are content selective, and I showed it in two different ways. First, that sharper ripple events themselves recapitulated the content preference that were observed during viewing. Okay, that the ripples during recall simply re-expressed the same picture preferences that were viewed during viewing. And second, that I showed you reactivation of content-specific visual representations in the cortex. This means that when you recall in faces, only the face selective centers in the cortex are coupled to the ripples. However, when you and not the place selective, and vice versa, when you recall places, the place selective representations in the visual cortex are activated, but not the faces. Okay, and this is another dimension, another way to show this content specificity of these events. And finally, I showed you some evidence that may suggest that I showed you that cortical activation slightly precedes the hippocampal ripple, and this is maybe compatible with the bidirectional signal flow between the cortex and the hippocampus, and at least not a unidirectional signal flow from the hippocampus to the cortex. Okay,
Just
so
before
now,
you continue, Yitzhak, there was a question in the chat from Yonatan
yes.
Katz.
Okay.
What is the cortical mean response during recall without ripple, if there are such events?
Great. So this is actually what I analyzed in this third study. So we're going to talk about it. However, I'm normalizing all the activation here are obviously normalized to this window, okay, to this window around the ripples. So any like global changes in the excitations of face areas and place areas during recollection are normalized out, and this is the response on top of this. But you are right in this question, and you are right in this question. And there is something very interesting happening in this visual cortex, even regardless of these ripples. And beside that, if you just time lock the response in, let's say, face selective areas to the onset of recollection, you don't see this response, okay? You see just scattered, very scattered, like jumps in activity, but you don't see any very clear response that you time lock the onset of recollection. Only when you align it to the hippocampal events, then you see this reactivation more clearly. You see, of course, some clue to this, because obviously it happens in the same times, but it's much more explained when you align the activity to the hippocampal ripples. They have some, yeah.
Just a comment about this point 4, that you know, the bidirectional that people have shown, you know, in rodents, if you look at the ripple-triggered PSTH, ripple-triggered spiking, yes, of hippocampal neurons, then they actually, you know, they're spread, and spiking starts to elevate much earlier, easily 200, 300 milliseconds before the ripple. By the way, we've seen this in bats as well. So then you could, I mean, it actually looks very similar to the red thing here. So it could be very easily said that the first spikes, the spikes that were elevated already before the ripple, which are the ones that are transmitted to the cortex, eventually it's the spikes that matter, they can be unidirectional. So that just to say that this whole bidirectionality is hard to defend. I think we can discuss it more at the end, but
okay.
Ripple, yeah, the elevated firing is prolonged.
Yes.
Menachem, you wanted to say something?
Actually, yes, you just said it. The ripple itself is about 50 milliseconds. And in some traditional work, it looked like the ripple starts the activity in the other areas in cortex where hippocampus presents. CA1 presents to subiculum, so that's another synapse. But what you are showing is that everything starts way before the ripple.
Okay.
In the cortex.
So, you know, because this question comes up again and again, maybe I will show you just some further results. So first of all, just to answer the Yonatan Katz question. So here you see the response just aligned to the verbal onset of report, okay? In the visual category selective areas. So you see the preferred category, the non-preferred category. So you don't see any clear activation. You see just this very jumpy activation here for the preferred category. Only when you align it to the hippocampal event, then you see a clear activation of these electrodes. So this is one thing. Second, and this is, I will go back to this, sorry. Sorry, yeah. Again, sorry. What is it? I don't want to spend too much time. Okay, here. So what I did is I also ran a multivariate pattern analysis, okay, on these cortical sites. So I took all these content-specific cortical site, high-order cortical visual sites, and now created these multi-site patterns for each one of the items. So you see here items 1 to 28, and all these spaces, all these places, I ordered the electrodes according to their phase selectivity. You see here. And now after dimensionality reduction, I have these very nice patterns that differentiate between spaces and places, but also between capture some of the variance across items. And now with these patterns, went back to the, I'm doing the following analysis. I'm taking the patterns that emerge during picture viewing, okay? And the patterns that emerge during recollection relative to the sharper ripple onset, okay? And now I'm computing the Pearson correlation between these patterns during viewing and patterns during recall. And all of this is relative to the recall onset. So just to explain this figure, what you see here is the correlation between patterns between recall and viewing relative to, in the x-axis, time from ripple onset, and in the y-axis is the time from picture viewing on the screen. And what you see here is a transient increase in the correlation, in the similarity between these patterns that is coupled, that is selectively emerges at the onset of the ripple, you see here. And here you see that it's much less, it happens much more precisely around the ripple onset. Okay, and the ripple onset is not the actual beginning of the ripple, just when it crosses 2 standard deviation, but the event started even slightly before that. So you see that overall it's rather coupled together, simultaneous. And second, I did the classification analysis, and I tried to decode based on the patterns that are emerged during recollection. I tried to decode and look at this panel here. What is the exact picture that the patient is retrieving? Solely by looking at the similarity to their, by training the classifier on the viewing pattern. So this is a cross classification analysis. Okay, I'm training on the viewing and then testing on the recall. And you see that a significant performance of the classifier emerging just, you see at time point 0, slightly before that, but time point 0. And compared to the category coding, you see that category can be decoded even before that. So this is another demonstration that when talking about the information itself, about the representation, not about the information is more sharp, more precise at the time of the ripple and not before the ripple. Okay, so
just a small technical question.
Yes.
Why did you need to do the PCA? Why couldn't you do the correlation on the original data?
Because the signal, first of all, the PCL removes a lot of noise, and you may have one noisy electrode that governs and dominates the pattern. Okay, so PCA just avoid everything this, and it's the right thing to do for classification analysis error, I think. But can talk about it.
Just to mention that it's reminiscent. There was a study in monkeys in IT cortex showing that the kind of responses to category for faces appeared earlier than the exemplars. There was this paper from 2000 or something like that. So it sort of goes along the similar lines.
And anyway, I think sorry. And another notion that you can take from it, which is my interpretation, but you see the fact that category information is activated even more before the ripple may also suggest that this information here perhaps bias the activity in the hippocampus that to reactivate the relevant representation and not, for example, representation from another category. We will talk about it in the
Yitzhak, I recommend to proceed.
Yeah, let's go on.
Yeah, sorry. So now quickly, I want to move to the next study. Now we wanted to generalize the result that we have on this episodic recollection also to the full range of declarative memory in humans, and also to remote autobiographic and semantic recollection. And to do that, we ran the following experiment. The patients were presented with short statements on the screen that you see, and they needed to do a true-false judgments on this statement. Now we had 4 different conditions. First, we have these statements such as "I wore jeans yesterday" that require the patient to do an autobiographical retrieval in order to answer them, in order to judge them. So I need to recall what I wear yesterday, and then I can say true or false. Okay, and as a control, we had a condition of math, of arithmetic calculation, and also we had the resting state condition, where the patient simply fixates on the screen for 5 to 10 seconds. And in a subgroup, 11 subjects, we also had semantic facts that actually require the patient to retrieve semantic knowledge. And the autobiographical statement, importantly, were spanned 4 different levels of remoteness. Okay, either statements that refer to events from today, events from yesterday. For example, I had chicken for dinner yesterday. I used my credit card this week and last month. Okay, so these 4 categories of memory age, okay, they will be useful. You will see what they tell us about the memory age and the ripples later. Now, for the main analysis, so again, I'm starting with the results from the hippocampus. And this time here in these patients, we had multiple recording sites in the hippocampus. Okay, so this means that we could now analyze a very exciting signal of multi-site SWR activity patterns, and not merely looking on one single recording from the hippocampus and on the local ripple rates there. And this will be very interesting later. And next, after that, I'm going to present a more, the hippocampal cortical interplay, but now taking much broader view with much more electrodes in the cortex, and you will see also there an interesting effect. So here you can see an overview of all hippocampal electrodes from the 20 patients that we had. Total of 112 electrodes in the hippocampus. We see beautiful ripples in all these recording sites. We included only recording sites in the CA1, CA2, and the subiculum, all the hippocampal output pathway, basically that display this ripple events. And the overall ripple rate across the task was about 0.4 events per second. This is very compatible with other human studies and studies in rodents. And now the main analysis that we did is the following. We simply computed the ripple rate across all experimental conditions, across every experimental condition. And what we see is the following. We see a highly significant increase in ripple rate for the autobiographical trials, compared both to the rest condition and to the mathematical calculation. And very interestingly, we see a significant reduction in ripple rate during mathematical calculation compared to rest. And this is, it's a very interesting result, because it shows you that when the patient is required to do a very demanding cognitive task that does not require long-term retrieval, then the hippocampus shut down or slightly down-regulates these ripples again. And here you see the sharper ripple density plot. Okay, so what you see in the y-axis is simply all the individual trials pulled together across all electrodes. Okay, and here you see the time from stimulus onset of the screen, and the trials are sorted according to the reaction time. And you see here all the autobiographical trials and all the math trials. Okay, and what you see is that ripple rate is increasing, indeed increasing, and it nicely tracks the reaction times in this trial. So next, what we did is we computed the peri-ripple time histogram of peri-event time histogram of ripples aligned to the reaction time of the patient. Okay, and what you see is a significant increase in ripple rate that peaks about 700 milliseconds before the patient is ready to do the judgment, which means, I think, that it reflects the retrieval of the memory, and then the patient is ready and doing the judgment. So we see this significant increase for the autobiographical trials and decrease for the math trials. And next, what we did is we went to the subgroup of 11 patients that had the semantic condition. And what we saw there was quite surprising for me at the beginning. We saw that semantic retrieval produces just as many ripples as the autobiographical retrieval. Okay, so it's not selective to autobiographical retrieval. It actually pertains to both kinds of declarative memory in humans. And here you see the peri-event time histogram of autobiographic and semantic statements. And now, given this result, we went on and now zoom in a little bit to the individual contact level. Okay, and what we found was that some of the electrodes show some preference to autobiographical statement. Other electrodes some bias to the semantic statement. However, you see here that their distribution across the hippocampus, and you see that it's rather uniform and represents some kind of mosaic organization. And this told us, or this calls for a multivariate analysis to take a slightly different approach in analyzing this data and look on the multi-site Shapur ripples patterns. And so we move from univariate analysis of simply averaging all the ripple rate across this electrode to looking on the patterns of multi-site SWR activity. And what we found there was very interesting. Okay, so first of all, we ran a LDA, a linear discriminant analysis classifier on these patterns of Shapur ripples. And what we find was that when you look on the patterns of hippocampal ripples, we see in the single trial level, in the single item level, we see ability to discriminate between the two memory types, significant performance of the classifier to discriminate between autobiographical, semantic, and also the mask trial, obviously. And you see here the shuffled data, okay, when you shuffle the labels of the trials, and you see that we get a highly significant performance of the classifier compared to the shuffling. And here you see the confusion matrix. So you see that the classifier was pretty accurate for the autobiographical, the math conditions. For the semantic condition, it's slightly confused between the autobiographical and the semantic. But this shows that at least the patterns of Shapur ripple activity across the hippocampus is highly informative about the memory content. And with these informative patterns, we now move to ask very interesting question about memory semantization. And what I mean by memory semantization, so think about it, but every learning experience that we have in life begins as an autobiographical event memory, okay? I remember myself in school, I don't know, in the elementary school, going with my geography teacher to the garden, and then she brings this atlas and showed me where Ireland was, okay? So I have this autobiographical event memory that as time passes, slightly fades away, transforms, become a bit more abstract. And then eventually, it becomes a semantic memory of just the relations, you know, between the concept and where is Ireland, where is the United Kingdom, etc. So now the question is, what is the process in the brains that reflect this semantization of memories? And we had a great opportunity to actually try to see this process occurring in the hippocampus. And how we did it, just to remind you, we had 4 different categories of statements in the autobiographical condition that explored 4 different levels of remoteness of autobiographical events. So we had events that occurred from today, events that occurred yesterday, events that occurred in the last week and in the last month. These are the oldest memories. These are the most recent memories. And we also had the semantic facts, which are in fact much more older memories, months to years back. Okay. Now the question is, how the ripple patterns look like for each type of memory, and what is the relation between these autobiographical patterns and the patterns associated with semantic memory? And this is the analysis that we did. We computed the distance between the patterns associated with a very recent memories and memories from yesterday, memories from last week, from last month. And what we see is this very nice gradual decrease in the distance from the average pattern of semantic memory. And this gradual change, decrease in distance from semantic memory may reflect the process of semantizations of memories, of an autobiographical memory, slowly becoming more abstract, until eventually it remains only as a semantic memory. And I think this result is interesting because it shows for the first time how this activity actually occurred already in the hippocampus. And this transformation from episodic to semantic is also related to sharp wave ripple activity across the hippocampus.
Yitzhak, one question about this. You say it yourself in some words, but it also puts a limitation on the interpretation. You interpret it as a matter of obstruction, and my question is, maybe it's only a matter of obstruction and not of time, because I mean, maybe it was an example, and so the question is, what you meant, this was medicine, this was a dinner, this was, I don't remember, went to a wedding. Did you try? Did I go to a wedding yesterday? Did I went to a wedding? Did I go to a wedding yesterday? Because I mean, here they're very correlated, the level of obstruction and the temporal domain.
So I cannot, I can tell you that I cannot disentangle the two. Obviously, the only manipulation that was made was on the memory age, either referring to events that happened yesterday or 2 months ago. And so this remains for future research to determine. However, I do see also a change in ripple rate, in the univariate change in ripple rate, also that reflect the memory age, but not the semantization. And perhaps we can maybe leave it for later, just because I don't have enough time. I want to show you some more. Okay, this is a valid point, and we need to discuss this.
Related to this question, can I add to that? Could it simply be a level of the richness of the memory, meaning semantic memories are poorer, and while memories that happen today are richer in content? So it's not a matter of semantization, but rather degradation of the quality of the memory in a way?
Completely, yeah, I completely agree. So maybe, yes, maybe when I'm saying semantization, I should say, semantization versus also abstraction, maybe also maturation of the memory, also fading away of the memory. All of these are possible interpretations for this result. However, still, this gradient raises some interesting questions about the process in the hippocampus. And yeah, but it leaves a lot of also open questions.
Yeah, but my math knowledge should be abstract and old as well. So it's kind of that's what makes it ambiguous, the interpretation. Okay.
Agree, but also I also showed you that math processing actually reduces some of the ripple activity. So maybe the patterns there are also a bit more like less, I don't know, formative.
I wonder also if there was an inter-subject attitude difference in the attitude to math, meaning math can be probably seen differently by different people. Right?
Interesting question. So I actually, no, I don't have this, but I just have this, maybe I just have this single units data. Here you see the data points. So indeed, I never looked at it, but 3 of the patients, of the 20 patients, actually showed a bit higher ripple rate. But I don't know, maybe it's the one that's here. So I don't have this question, but it's an interesting analysis to do whether there is some differences between patients. Okay, so finally, I just want to touch one last thing about this project, and then quickly run to the third project. So just to remind you, I had many cortical electrodes in these patients as well, okay, covering a major portion of the cortex. So now I wanted to see what is the peri-ripple responses across the entire cortex, okay? And to do that, I ran a deconvolution analysis by fitting a general linear model to the broadband gamma responses. Now this model, using this analysis, I'm modeling both the stimulus events, the patient's responses, and the hippocampal ripples all together, and fitting. And when this model is solved, I'm actually taking into account all events together, and this allows me to isolate the peri-ripple response, even though it's slightly overlapped with the other responses. Okay, so because these responses may overlap in time in this design, then we needed to do this deconvolution. However, it works really nicely, and we have some control analysis that's showing that it's indeed the analysis does what it's supposed to do. But what we have in the end is the peri-ripple activation across all these cortical electrodes. Here you see the most example from 2 electrodes, the posterior cingulate and the superior frontal side. And now for the group level results, you see here, this is the map of the magnitude of peri-ripple activation across the cortex. And what you see here is that in medial regions of the brain, and also in the prefrontal and the orbital frontal and the superior frontal gyrus, you see a very pronounced peri-ripple response during the recollection. And taking a more data-driven approach, you see here the entire map. And I plotted all the electrodes that exhibited a prominent peri-ripple response. And if you can see also the pink areas in the background, you see that actually the strongest peri-ripple responses overlapped with the regions known to be related to the default mode networks humans, which this is a very large-scale network that was shown in fMRI studies to be involved in autobiographical recall. And we see that indeed the peri-ripple responses in the cortex during retrieval overlap with the default mode network. You can see it here in this analysis. So you see we compared the peri-ripple responses across the default mode network and the several other large-scale networks in the cortex. So you see here, this is the default, the ripple triggered response in the default mode network, and compared to all other cortical networks. And okay, and that's it. Now, just to summarize what I showed you in this part, I showed you that ripples are elicited during both recent and remote recollection, and during both episodic and semantic declarative memories. I showed you that ripples patterns reflect both memory content and the perhaps semantization or abstraction of these memory representations in the hippocampus. And I showed you also that a prominent hippocampal default mode network interaction that is coupled to these ripple events. Now, finally, maybe I will just run through this last part, which we ask slightly a different question. Now we focus on the cortex, and we ask, how does the brain set boundaries to spontaneous recollection? And to demonstrate this question, let's now try to freely recall some, let's say, male movie actors that you remember. Okay, so when you try to do this task, you can easily think about names such as Brad Pitt, Tom Hanks. Maybe after a while, you also come up with Leonardo DiCaprio. But interestingly, you will not come up with names such as Bill Clinton or Leo Messi. How is that? How does the brain imposes predefined boundaries on our next recollection? How does it control which category are going to be the new memories that are going to appear in our mind are going to belong to? So this is a very deep question of how in fact we control this retrieval process. And so far I showed you that the hippocampus play a major role in eliciting spontaneous recollection, or perhaps in the emergence of spontaneous recollection. Now I want to show you is to examine whether what is the role that the cortex have on setting boundaries to this spontaneous recollection. And we know that the prefrontal cortex, for example, holds representation of the goals of the retrieval. However, however, what is the precise mechanism that allows the cortex to actually influence what happens in the hippocampus during a individual recollection? This is an open question, and I investigated it using the same task that I showed you in the first study. Okay, so here, just to remind you, the patients were required to retrieve the picture that they have seen in two separate blocks. First, focusing on the faces, then targeting all the places and trying to avoid recalling faces. Okay, so this is a category selective retrieval. And now we ask what happens in the cortex, again, in these very unique electrodes in the inferior temporal cortex that are selective to only one category. And what we found is the following. We found that throughout the free recall period, there was a slight but consistent and persistent increase in the baseline activity level of these electrodes, only when the patient recalled, their preferred category. So when the patient recalled places, this electrode remains around the baseline level, and only when the patient recalled faces, you see this slight increase in baseline activity. And you see it also vice versa. You see it in place selective electrode. You see a slight increase in the baseline activity of this cortical sites when the patient's retrieving places. And now here is the group level analysis. You see a significant interaction between face selective electrodes and place selective electrodes. And you see that face selective electrodes slightly enhance their activity during face retrieval. And this is the median activity of each electrode, average across all electrodes. And now the question, okay, so we see an increase in median amplitude, an increase in median amplitude in category selective regions. However, maybe this increase in activity is simply a result and is simply led by the visual reactivation associated with individual recollection. To rule out this option, we did the following analysis. We simply excluded all time points around individual recall events and analyzed only the inter-recall intervals, where the patients were rather silent and attempting to recall a particular category, but failed to come up with particular items. And we see there the same interaction, significant interaction. And next we ask, okay, so you see this increase in median amplitude, but is it a real baseline shift or simply accumulation of multiple transient activations in the cortex? So to rule this, to test this thing, what we did is we analyzed the distribution of momentary HFB, momentary high gamma amplitude in the cortex. And just to illustrate it, we have two opposing models. Okay, so one says that perhaps the hippocampus, the cortex elicits this transient burst. The other says that the cortex slightly enhanced the baseline activity. Now to differentiate between the two models, we plotted the distribution of cortical of amplitude values, and what you see that the two models differs in the following manner. When for the burst model, you see a slight increase when you add this transient burst. You see an increase in the right tail of the distribution of amplitude values, and if indeed what we have is a baseline shift, then you expect to see an entire shift of the distribution to the right. Okay, and this I think visualizes the two predictions better. So here you see what happens if I'm adding to the signal burst during 10, 20, and 30% of the time points. So you see a non-symmetric increase over the top percentiles of the distribution. And if you add a constant baseline shift to the signal, then you expect to see a comparable increase across all percentiles. And now for the real data, what we see during picture viewing, we see indeed a burst model, that the burst model is correct, is more representative of the signal. Okay, we see an increase, a higher increase for the top percentiles. And during free recall, what we see is indeed something that is much more compatible with the baseline shift model. Okay, we don't see an increase, a higher increase in the top percentiles, and we see a comparable increase across all percentiles. We can just to interpret these results. So what we see is that during recall, probably most likely the prefrontal cortex generates some signal related to the goals of the retrieval, which is then translated through attentional modulation mechanism to a slight increase of activity in category selective representations in association cortexes, which then converge on the hippocampus eventually. And just to demonstrate this process from the beginning, and this is the last thing I'm going to talk about, imagine that you are trying to recall faces. So now you increase the baseline activity in face selective sites in the cortex. And with this increased level of activity, what happens in the end is that the hippocampus inputs to the hippocampus are slightly biased toward faces, perhaps facilitating representations in the hippocampus that are related to faces. And then the hippocampus elicits a memory representation associated with ripples. And this broadcasts this information back to the cortex of a particular item belonging to the desired category and not to the other category. Okay, so this is what we believe happens. And just to summarize the last part, everything that I said to you, so I showed you a role for hippocampal ripples in declarative memory recall in humans. Ripples were elicited during both recent and remote memories, episodic and semantic. And I showed you sharper ripple. I suggested sharper ripple as an orchestration mechanism that coordinate the cortical activation related to the memory representations during recollection. And I showed you that sharper ripple activity patterns in the hippocampus reflect both the content of the memory and perhaps a semantization process in the hippocampus. And I showed you a cortical mechanism that may account for the remarkable flexibility and ease by which we constrain our recollection to a particular context or semantic field. And this is by baseline shifts that modulate the baseline activity in different cortical sites during recollection. So, okay, so with that, I want to end. I want to thank, first of all, my collaborators, our collaborators, the lab of Professor Shesh Mehta in New York, in Long Island, and the group of Professor Joseph Parvizi in Stanford University. And I want to say a special thanks also to the patients with that kindly cooperated and participated in our studies. And I want now to also thank my mentor, Rafi. And I want to take this opportunity to first of all, thank you for really for your dedication as a mentor, and for the support and the constant encouragement that you give to your students, which is something really not obvious. The fact that no matter when, every email, every question, every paper draft that I send you, within 24 hours, I'm going to receive it back to my email. And you know, this is really keep the enthusiasm and keep the workflow and the fire. And thank you for that. And I want also to thank you for, you know, all the little conversations that we had, which were always, you know, full of curiosity and excitement and philosophical argument, and were pretty loud sometimes, but really it was fun. And
so that's why Rafi closes the door. He shouts at you, and then, oh, now I understand.
All the time we
All
shout,
in good heart.
and this is just because we really enjoyed, you know, arguing about this science. And
of course.
And also, I want to say that that, you know, you give your students with the confidence, you know, and the courage that is really needed for doing some independent thinking and do this research, and it's super important. And I thank you for that. And you also teach us how to look at the data, you know, and simplicity and clarity. This is your main rules. And you really, you showed me how to, you know, look at the data and see what the data tells us, and not to, you know, follow what other things or what should be the case, but what actually the data says. And I'm going to take these lessons to my next chapter journey. So thank you very much.
Thanks, Yitzhak. Very moving.
I want to thank also all the lab members, and especially Michal Harel, which really our lab manager, which always just wanted to offer her help in any way possible. So thank you, Michal, and thank you all the other lab members and my friends from the department. And finally, my family, which supported me, you know, throughout all this work. My wife, my best friend Noah, and my 2.5-year-old daughter Sol. So with that, I want to end. Thank you very much. And now we have time for questions.